What we're going to do now is we're going to have three presentations from trainees. And the first trainee who's going to present to us is Erik Lamoureux, who works in the laboratory of Hongshan Ma, who you'll also be hearing from later today. And Erik is going to give us a fairly brief summary of what he's been up to. So, Erik, over to you.
Great. Thank you, Dana. Hello, everyone. My name is Erik Lamoureux, and I'm a mechanical engineering master's student in the Ma Lab. Today, I'm discussing red blood cell deformability classification using deep learning. Now, moving on to red blood cell deformability. Next slide. This deformability is an important biophysical property that determines the cell's ability to squeeze through and navigate the body's microvasculature. Transfusion red blood cell deformability varies based on both the donor and the blood storage time. The sorting of blood based on deformability would allow for patient-specific allocation of transfusion blood, where for chronic transfusion patients, they should receive long-lasting or more deformable blood to increase the time between needed transfusions. And for acute injury patients, they should receive short-lasting or more rigid blood, as they just need a blood top-up and can normally produce an adequate blood supply. Here, I present a machine learning approach to determine blood deformability that is accessible to anyone with an adequate optical microscope. Next, I will discuss the general approach. Blood was acquired from a donor and was run through a microfluidic device at 1 to 2% hematocrit. Shown here in the enlarged image, the microfluidic device sorts cells based on their deformability by squeezing them through progressively narrower constrictions along the upward flow trajectory. When the cells can no longer pass through the constrictions, they are then sorted to a distinct deformability outlet. More deformable cells travel further along the upward flow trajectory to lower numbered outlets, while more rigid cells do not travel as far and are sorted to higher numbered outlets. After the sample is run for 1 to 2 hours, optical microscopy images were acquired and segmented into single cell images. Balanced classes were created consisting of 10,000 images each for training and 2,000 images each for testing. The convolutional neural network was trained using the training dataset and tested using the testing dataset. Finally, the machine learning classification results were compared to the microfluidic sorting results. Now moving on to these microfluidic results. Next slide. Yeah, so here shows the results for donor 3 at day 1 storage. In the top left shows the normal distribution of cells sorted to outlets, and there are enough cells sorted to outlets 3 and 4 for a database creation. To the right of this is the cumulative distribution plot with the 50% crossover frequency shown, and this is defined as the rigidity score of the donor, which is 3.27. After microfluidic sorting, 40x magnification bright field full image scans were obtained using a Nikon microscope, and this is shown on the bottom left. These images were then input to a custom segmentation algorithm to extract individual cells into new images. Then these cells were augmented using a random multiple of 90-degree rotation, which removed well location shadowing and created balanced classes. The training and testing images came from separate wells in a well plate to ensure independence between training and testing data sets. Once the databases were created, they were input into a deep learning model, and this is abstracted on the bottom right of the page. Broadly speaking, 60 by 60 pixel images were passed through a convolutional neural network, which learns important features that differentiates the images based on their cellular deformability. The data set of 20,000 images total was run through the network 50 times to learn the deformability differences. The validation accuracy generally converged to the training accuracy, but some variation remained, and this is shown in the bottom right corner. Now moving on to testing results. The model here obtained 98% training, 96% validation, and 86% testing accuracies. In the confusion matrix on the left of the page, the accuracy of outlet 3 classification was 98%, which was far better than outlet 4 at 75%. Even with this difference, the 50% crossover frequency or rigidity score was similar between the two methods. and there was strong and significant correlation between the machine learning and microfluidic distributions. The rigidity score difference of 0.34 is near the natural variation of a donor's rigidity score, indicating that this machine learning method classifies red blood cell deformability similar to the microfluidic device. Next, I will discuss future directions. I aim to improve this method by improving the sample preparation by fixing red blood cells and staining them prior to imaging, and this will provide fewer image artifacts. I want to test the method on a variety of donors and storage times to classify between common outlets, such as outlets 2 to 6. I look to train the model on one donor and test on another to provide inter-donor generalizability. And the ultimate goal here is to create a machine learning pipeline that takes an unsorted image, possibly from a blood smear, to determine any donor's deformability profile with an anticipated image result of this method shown to the right. Thank you.
Thanks very much, Erik. We have time for a question or two if people have them, and you could, again, put your questions in the chat box, or you can take yourself off mute and ask them directly.
I'll jump in. So Erik, that was great. A question, is deformability entirely intrinsic to the cell, or like when you apply it through the microfluidic device, does it vary in, how do you apply pressure for it to go through the microfluidic device?
Sure, so there's a couple pressures that are applied to the device. So there's a forward pressure that sends the cells through the sorting matrix. And there's also up and down pressure that alternates, but alternates at different frequencies. So the upward flow pressure goes for 5 seconds and downward flow pressure goes for 1. And so the upward pressure pushes it through the constrictions, and the downward pressure dislodges them if they cannot travel through the constrictions.
So those conditions have to be consistent through.
Yeah, so there is a electrical, mechanical, integrated setup that monitors that and keeps the pressures constant throughout.
Okay, thanks.
Erik, there's one more question in the chat room, which is, do you anticipate that fixation will change your red cell morphology?
Yeah, so that's one thing that we're going to look at. We know that by fixation you kill the cells, and that might affect how these images, how well these images can determine the deformability and how relevant that is to the live cell and how it is sorted through the device. But it is, if it's consistent, the morphological change in the killing of the cell, we still may be able to determine the deformability purely from the images relative to the microfluidic device. And this is morphological deformation, membrane deformity, or oxidation. How I would describe it is just how well the cell is able to squeeze through narrow constrictions. So I guess that would be the membrane deformability. And the microfluidic device is meant to mimic the sorting filters in the spleen or the kidney. And so we're using the images to compare directly against this physical test to see if just taking images of it can give us the same results. Yeah.
Okay, great. Thank you very much, Erik. Really nice talk.
Thank you.
We're going to move on to our next speaker, who is Niloy Chatterjee, and he's coming to us from the University of Calcutta. So hopefully,
hello, ma'am. Am I audible?
Yep, there you are. We can hear you. Please
Okay,
go right ahead.
Good morning to all the teachers, professors, and distinguished speakers, and all the audiences present here. I am Niloy Chatterjee from University of Calcutta, Kolkata, India. I am a research scholar working in that university. As nowadays we are in a catastrophic situation being due to COVID-19. So my topic of talk is how blood matters in this COVID-19 situation or infection. We all know that blood is a connective tissue which transports substances throughout the body. So now to start with the infection cycle, it is seen that blood clot is an important phenomenon in this infection. As after COVID infection starts, then there is an initial inflammation, due to which after that there is release of inflammatory proteins, which alters the blood platelet function, that results in the hyperactivity and hyperaggregation of the platelets in the human body. This leads to overactive blood coagulation. Now, to go deeper into this, a study by University of Utah scientists have go depth, and they found that what are the mechanisms which regulate these unnatural functions of platelets in the human system. So they found that COVID-19 proteins interact with the megakaryocyte or platelets precursor. Thus, they alter the structure of the platelets. Again, as a they also indirectly changes in the genetic way of the platelets, though no direct links have been established. Again, there has been inhibition of the platelet activation, which is required for the proper functioning of them. This leads to the hyperaggregation and the interaction with the immune system, such as in lungs, that are hugely altered. The combination of all these factors leads to hyperactive blood clotting, which ultimately leads to multi-organ failure, inflammation of lungs, respiratory tract, and cardiovascular problems. It is also seen that if any anti-inflammatory medicines, such as aspirin, which is also a blood thinner, if given to the patients, then these complications can be averted. Next slide, ma'am. Now coming to the question that whether this blood clotting is a boon or a curse for us. Most of us would say that it is actually a curse, but this is not the situation. After COVID-19 infection, it leads to inflammation. Now this inflammation and coagulation moves in a bidirectional manner, which are regulated by well-defined biochemical signals. That leads to cytokinemia, means that overexpression of cytokines, where pro-inflammatory cytokines are secreted like IL-6, IL-1 and TNF-alpha. These cytokines control thrombin activation as well as anticoagulant activity. But in COVID patients, these are not regulated properly. So excess thrombin is released, which results to microthrombosis, that is small micro blood clot formation in various parts of the body. So COVID patients have been found that they have about 7 to 8% lower concentration of antithrombin in the body. This leads to thromboembolic complications, that is blood clots in various vessels which break loose and are carried to other vessels and they block the passage of blood. This is also known as disseminated intravascular coagulation. If we look at the other way, that is in the upper pathway, after infection there is a polyphasic immune response. That is the overall immune response changes in magnitude from time to time, finally leading to overall immunosuppression in innate and adaptive immunity. Sensing this, there is pulmonary thromboembolosis and cytokine storm, which ultimately leads to death. Okay. Now, however, blood clot can be prevented, and this blood clot is either good or not. That is a matter of debate, as it also aids in recovery, and to stop the spread of the virus, this blood clot is thought to be a good mechanism. Now, in the next slide, we have no biological biomarker available till date to know if COVID-19 is susceptible in a population or not. So we have some authors have in a study have tried to see whether blood grouping or ABO blood grouping system is related to that or not. It has been found in by studies in Italy, Spain, and China and Hong Kong that A blood group are more susceptible to comorbidity as well as serious respiratory distress and other complications of COVID, while O is at lesser risk. Antibodies are previously found to inhibit SARS-CoV antibation, not SARS-CoV-2 to ACE2, so they might play a role. Besides that, Rh positive people are at higher risk. The antigen polysaccharide composition in RBC, anti-blood antibodies may influence the virus surface structure and therefore susceptibility. Besides, blood proteins may also affect the immune response to individuals. Genetic studies showed that SNPs in the 9th chromosome, 9q34.2 position, shows huge variations in COVID-19 patients. This position coincides with the ABO blood group loci, and in that loci there has been a gene SLC6A20, which is associated with ACE2 receptor. Besides, however, it can also be said that recent studies at Harvard and Massachusetts found no association between blood group and COVID susceptibility. Next slide, ma'am. We all know that for the real-time reverse transcriptase PCR is the gold standard for the diagnosis of COVID-19. But there are some drawbacks of that. It requires long turnaround time, it has less specificity, and it is a little bit costly. So we can shift to other testing methods like blood or serological testing, which can be good alternatives. The current alternatives which are present are rapid diagnostic tests, which is very fast, but they provide qualitative estimation of antibodies. ELISA provides quantitative estimation, but the real amount of antibodies which can inhibit virus growth, it cannot be known. Third is neutralization assay, which is time consuming, but in that we may not know which protein or viral proteins are involved in replication or which are inert. And finally is chemiluminescent immunoassay, which is quantitative in nature, but it also cannot show us that which can inhibit virus replication or not. So some studies have been conducted to see if blood parameters can contribute to help us know whether a patient is positive or not. In a study, this has been identified that some parameters like WBC, neutrophil, lymphocyte, and platelets go down drastically, which are statistically significant in COVID-19 patients, while ALT, AST, GGT, LDH, and C-reactive protein grow up many folds. So these can be used as parameters to distinguish between COVID positive and COVID negative. In another study recently in Spain, the group have used machine learning and artificial intelligence to use to see whether they can be used for serological testing or not. They identified 11 blood parameters like total protein content, bilirubin content, glucose, creatinine, calcium, creatinine kinase, LDH, potassium content, magnesium, platelet distribution, width, and basophil count. They have used the deep neural learning and random tree algorithm for their calculation. So it can be seen that if we want, then we can use blood as diagnostic tool also for this disease. Next slide, ma'am. As we know that in case of COVID-19, there has been no current vaccine or therapy are available. So blood can be also used as a therapeutic target. Convalescent plasma therapy can help in prophylaxis as well as treatment. Previously, it has been used in previous Spanish flu, polio, Ebola, rabies, etc. Recovered patients actually develop circulatory antibodies to SARS-CoV-2 proteins, which are protective in nature, and they can also neutralize future infections from SARS-CoV-2. Patients have been seen to have lower mortality, clinical improvement, but the role is still not yet clear. In China, Netherlands, and Iraq, it has shown that convalescent plasma therapy is improved. But while in some other countries like Spain and some other studies have been conducted, even today in my country, ICMR has published a study that convalescent plasma therapy is futile and it cannot be so much be trusted for the therapeutic target. In Kolkata, scientist Dippoman Ganguly of Indian Institute of Chemical Biology is currently working on a small-scale clinical trial of this. So it's a matter of debate that whether they are actually magical bullets in the time of these pandemics or not. It is a matter of subject of debate. Next slide, ma'am. Finally, I think I've completed, and I'm open to any questions if anybody is.
Thank you very much, Niloy. We have time for one question.
Yes, ma'am.
And again, you can put your question in the chat box. Yeah, so maybe I'll just ask you briefly if you could just so you're linking all of these various systems, coagulation, the ABO group. So is the place where you see this coming together over the von Willebrand factor levels, where you see the important point of focus?
Ma'am, actually, we don't have any biomarkers to predict COVID-19. Okay, so if this blood group linkage is found prominent, then we can, I think that we can, at the preliminary stage, we can predict, or we can see that who are at the relative more risk of this developing this infection or not. Okay, and blood coagulation has been an intrinsic factor which is present in the COVID-19 from the start to the end, if we can see. So these two means phenomena are highly acclaimed and means very much interrelated to each other.
Okay, thank you very much. All right, I think we'll move on to our third speaker, who is Amy Wong Strilchuk from the from Christian Kastrup's laboratory.
Hi, yeah, thanks for that introduction. Great, got the slides up. It's my pleasure to speak to you all today, wherever you're coming from. Following is just a short presentation, about the research I've been doing in the Kastrup lab here at UBC, namely using transient gene therapy, or siRNA, to knock down coagulation factor 13 to decrease the stability of thrombi for potential use in coagulopathy or thrombotic disease. Next slide, please. So starting from a clinical angle, as you know, therapeutics for coagulation disorders like thrombosis or the recent COVID-19 associated coagulopathy, as just talking about, are generally categorized as anticoagulants or thrombolytics. So anticoagulants inhibit the coagulation cascade, whereas thrombolytics promote, still on the last slide, whereas thrombolytics, promote the activation of plasmin to degrade fibrin. Both of these strategies have drawbacks, the biggest of which is the accompanying risk of bleeding. So a third strategy and possible solution to these limitations is to enhance the susceptibility of clots to endogenous fibrinolysis. And this could be done by inhibiting coagulation factor 13 to stop cross-linking of antifibrinolytics to clots. So as a review, factor 13 consists of two different subunits. The B subunit in green there holds the factor 13 complex together and extends its half-life in blood, whereas the A subunit is the transglutaminase that cross-links fibrin and antifibrinolytic proteins together to make clots resistant to fibrinolysis. So now, next slide, please. So researchers have been working on this idea for a while, but inhibitors for active factor 13A still do not meet the criteria for many long-term studies in vivo. So instead, we are taking a new approach in using siRNA to knock down the B subunit of factor 13. Because the B subunit is integral to the stabilization of the A subunit in circulation, factor 13B knockdown would destabilize the A subunit and cause its depletion from plasma. Factor 13B is a prime target for siRNA therapy. RNAi has been previously shown to be a potent modulator of coagulation. And in this work, we use a lipid nanoparticle to deliver our siRNA preferentially to the liver, where factor 13B is made. Next slide. So on the top left here, factor 13B mRNA levels were measured in liver tissue samples at weekly time points after the administration of a single dose of siRNA targeting factor 13B, and that's the green points, or a control nonspecific siRNA in purple. So as you can see, factor 13B mRNA was decreased by like 90% for 3 weeks after a single administration of the siRNA. And on the top right of the slide, factor 13A protein is quantified in plasma. So corresponding to the mRNA knockdown of factor 13B, factor 13A protein was depleted for 3 weeks post-administration of the SI, and repeat administration of the SI at 3-week intervals could sustain factor 13A depletion for months. Finally, on the bottom of this slide, a tail transection model of bleeding showed that while clots were less stable and prone to failing, overall blood loss was not increased compared to untreated mice because clots were able to form and reform quickly. Notably, this is different from the apixaban-treated mice, where blood loss was significantly increased. So next slide, please. In vitro, we confirmed that, sure enough, as was hypothesized, factor 13 knockdown made clots formed in vitro more susceptible to lysis, and this was because less antiplasmin was being cross-linked to clots. So on the top left here is a Western blot against antiplasmin, looking at the high molecular weight complexes usually formed by factor 13 cross-linking antiplasmin to fibrin. As you can see, there's a lot less high molecular weight antiplasmin in the SI factor 13 treated samples. Underneath, on the bottom left, are a couple of thromboelastography curves. These measure the strength or stiffness of clots as they're forming in vitro. So the green curve is the blood where factor 13 had been knocked down, and you can see that this clot lyses much faster than the control purple curve. Calling back to the results of the bleeding challenge as well, here in the TEG curves, there appears to be no difference in the clotting time in vitro. So finally on the right is shown an in vivo experiment whereby a vessel was occluded by exposure to ferric chloride, and tenecteplase, a TPA, was subsequently administered. At this dose of TPA, which is equivalent to about a half therapeutic dose, there was significantly more clot lysis and reperfusion of the vessel in animals pre-treated with SI factor 13. This reperfusion was quantified by measuring blood flow and graft below. So next slide. Okay, we're kind of running out of time, so maybe I'll just summarize by saying SI factor 13 represents a potent biological tool and potential future antithrombotic. This is just a summary of the results I just spoke about. And going forward, we have developed a similar reagent to knock factor 13 down in rabbits, and we'll be using it to study the application of this agent in ECMO. This is a clinical circumstance where patients are at risk of thrombosis, but at the same time at risk of bleeding. So it's a scenario where anticoagulation is difficult to manage. So next, I know I'm out of time, but I'd just like to end by thanking the many collaborators without whom this work could not be done. And I'll happily take any questions.
Great, thanks very much, Amy. So again, if people have questions, you could put them in the chat box. Maybe I'll just start off by asking you about what you anticipate will be the effect of the interference and reduction of B chain levels on platelet function, because you'll have an effect there as well as in the clotting system.
Yeah, so really good question. So yeah, I didn't mention this, but aside from the factor 13 that's circulating in plasma, there's also factor 13 inside platelets. Though this factor 13 is just a dimer of the A subunits. So the 13 that is made in platelets does not depend on stabilization from the B subunit. So that A should still be available and working in wound healing. There has always been a question of whether any 13 is taken up from the plasma into the platelets. So this might be disrupted, but I think the majority of platelet factor 13A would be fine, and we've measured that also.
Okay, thank you. Anyone have another question? All right, if not, thanks very much, Amy. That was really very interesting presentation.
Thank you.
Okay, and we will move on. So our next presenter is Dr. Karina Yazdanbakhsh from the New York Blood Center, and she's going to talk with us about some of her really very interesting work in transfusion immunology and sickle cell disease. She is the head of the Laboratory of Complement Biology at the Lindsay Kimball Research Institute of the New York Blood Center. And she's actually working very effectively in the sickle cell disease world. I just saw an announcement recently that she's got a huge bag of money out of the NIH to continue her research on hemolytic disorders in general and sickle cell disease in particular. Karina received her PhD in molecular biology from the National Institutes for Medical Research at Mill Hill in London, and then did her postdoctoral training in molecular and cellular immunology at Columbia and at the Rockefeller before she joined the New York Blood Center in 1996. So very welcome, Karina, and really happy to have you here finally. And I'm sorry that it's virtual and we can't go terrorize Vancouver. So.
Exactly. So hope you can hear me.
We can. Yeah.
Okay, Fantastic. So thank you so much to the organizers, especially Dana Devine, for the invitation. And after hearing Ed Conway's description of the person that this symposium is after, is totally an honor. And really, thank you so much for having me be part of this the symposium today. So what I like to do today is describe a couple of projects that are ongoing in the lab. And really the research focus of my lab has been, as it says, transfusion immunology, but really in the context of sickle cell disease. And what I want to really emphasize is that, and the work is going to show, of course, this is immune response to the transfused blood. That's what we're interested in. But really the recipient and, you know, the sickle cell pathophysiology, the disease background is going to be really important. It makes sense, it should be. But because of that, it's really some of these studies you'll see has gotten us into more and more trying to understand the disease pathophysiology before understanding how the transfusion is really intersecting with that. I mean, this is transfusion medicine, I know, but now we really, we're there, we are doing that. Okay, so I have no conflict. So what I'll do for the first half of the talk is to discuss the potential impact of hemolysis, which is a hallmark of sickle cell disease, on adaptive immune response and its role in alloimmunization in this disease, and then describe the role of a subset of circulating monocytes that we've uncovered to be important, we think they're important in the control of hemolysis-driven vaso-occlusion, another hallmark of sickle cell disease. So transfusions in the sickle cell, you know, therapy is critical for the management of sickle cell complications. We know that, for prevention of stroke, for treatment of acute chest syndrome. However, as we also know, there's a downside. The patient can make antibodies against allogeneic antigens on the transfused red cells. This can cause, in this patient population, really, you know, life-threatening transfusion reactions. It delays transfusions because you have to find the matched units. And so it's really puts the patient at risk of more severe complications. So of course, to prevent alloimmunization, it would be best just to do prophylactic matching. However, you know, the downside, the argument against it is that not all patients make alloantibodies, okay, when they're transfused, these patients especially. And at the highest estimate is like 50%, which is a lot. It's a lot of patients that make alloantibodies, but the other 50% don't. So it's too costly to do prophylactic matching. And potentially we may have problems identifying units that can satisfy all this population. They are in need of a lot of transfusions. So, and also another school of thought has said that the rare units should be reserved for patients who require these antigenic specifications. So what my lab and several others have been doing is like trying to get at the what we call the responder biomarkers, who, you know, trying to get at that so that we can predict in advance which patients will make antibodies, and really understanding alloimmunization risk factor, but from the immune side. And, you know, to begin to start to do that, we've taken the approach of really going into immune responsiveness of the patient. That's what we call it. We're calling it immune responsive. Why is it that some patients are doing that, are responders? Okay, so hemolysis, as I said, a hallmark of sickle cell disease. Red cell destruction can occur extravascularly and intravascularly. And in fact, an estimated 1/3 of hemolysis occurs apparently intravascularly in sickle cell disease. The excessive free heme can activate the underlying endothelium. It increases, upregulates the adhesion markers on the endothelial cells. There is increased attachments of leukocytes and the sickle red cells to this activated endothelium. And as a result, you see this blockage of the blood flow and you get the vaso-occlusion. That means there's not enough oxygen delivered to the tissues. The tissues start having failures, you know, they start dying. There's organ failure. So how do we know hemolysis is important? Well, if you inject free heme in mouse models of sickle cell, it induces vascular stasis and acute chest syndrome. We know that the heme scavenging removal system is overwhelmed, is shot away, is very low in these patients. However, this heme oxygenase 1, which breaks down heme, is upregulated. Another indication that heme is doing something in these patients. So what does this HO1 does, this heme oxygenase 1? Well, interestingly, it's not only it detoxifies the heme, but the breakdown products, basically carbon monoxide, bilirubin, they are have properties antioxidant, anti-proliferative, anti-inflammatory properties that they confirm onto the cell that they're produced in. And so and there has been this idea that HO1 could be tolerogenic. So we asked the question, well, what if you don't have enough HO, or there is something wrong with inability to process this heme? What would happen there? And being that is what happens in an immune cell, if you can handle the ongoing hemolysis, we reckon that that would be critical for the adaptive immune response, specifically alloimmunization. And this was really the start point of my talk, I have to say. So before I start any IgG response, you need T cell help, okay? Helper T cells, right? They are important for B cell help, for antibody production. So the first question we ask, does heme or its oxidized form hemine, I'm going to be using them interchangeably, alter T cell polarization? What do I mean by T cell polarization? Well, it's how a naive T cells becomes activated and takes on an effector function, effector property. So this happens, the naive T cells have to encounter antigen presenting cells. Dendritic cells are the most potent antigen presenting cells out there. They get, once they mature, they need toll-like receptors, that is like, because of the infectious agents that are coming in or the environment, you have sterile inflammation there, they get activated, they can become activated. That's when then they start talking to the naive T cell. And depending on the microenvironment there, let's say the type of cytokines there, they can take on different effector functions, we call them. So you can become a Th1 and you have a specific type of cytokine or transcription factors that can identify you. And so, at least with the Th1, it's kind of associated with more of a pro-inflammatory response. You can also become a Treg, which is more anti-inflammatory, right? It's keeping everything down, calming things down. So this is what I mean by T cell priming. So we ask the question, can these dendritic cells prime the T cells to become pro-inflammatory now in the context of hemolysis? Just to tell you how we do these experiments, the protocol is we isolate monocytes from peripheral mononuclear cells. We have this protocol, you get them to become dendritic cells, then you have to mature them, right? And the way we hit them with the TLR, toll-like receptor ligand, and we use different types, some are listed here, it's just a protocol, in the presence or absence of heme. And at that point, this is during the maturation, the dendritic cell becomes mature. And now we give them allogeneic naive T cells and ask, okay, how much Th1 cells do we get out at the end? And this is a healthy donor plot. What we see with increasing concentration of heme, you see a decrease in this Th1 priming that this healthy donor dendritic cell is instructing. So how about our patients? So we took some patients who are non-alloimmunized in blue and some who are alloimmunized. These are sickle patients. We've tried to be as controlling for the confounders as how many units they've been transfused, how long they've been on chronic transfusion, but this is what we have. So what we found was that healthy donors and non-alloimmunized, the blue guys, regardless of how we matured their dendritic cells, when they get exposed to heme, they downregulate their Th1. Okay, they say, don't get primed, don't just calm down, no. Whereas these alloimmunized patients, it was as if, so what, heme is there. This is, you know, no difference. And if we plot this change, what we found, as I said, the alloimmunized do not care whether there's heme around or not. They are going to induce Th1 responses. Whereas the non-alloimmunized, there's about 25% less Th1 priming going on. What is the mechanism? As I said, there is, you know, the maturation is very important. This is where we have it. So we looked at, does it affect dendritic cell maturation? And CD83 is a marker of maturation, so we focused on that one. And what we found with increasing, this is again you know, what we're looking at, the effect of heme on CD83 expression on these mature dendritic cells. It's as if with increasing concentration of heme, you have less of the CD83. There's less maturation. So it's almost down-regulating the maturation of these dendritic cells. And again, going to our just focusing on non-alloimmunized and alloimmunized, again, the alloimmunized patients don't care whether they're seeing heme, they have enough CD83 expression, they're maturing just fine in the presence of heme. Whereas the non-alloimmunized saying, calm down, do not get mature, do not, don't get mature. That's it. So it has been shown by beautiful work from Belcher and Versalotti that heme binds to toll-like receptor 4 and induces this pro-inflammatory oxidant pathway. You know, it goes through that. So we asked to ask whether this down-regulation of CD83 is occurring through TLR4. We use anti-TLR4, and just as well, you know, basically the data says that if you use anti-TLR4, block the TLR4, now heme, you do not get down-regulation of CD83, indicating that, and of course this is in healthy donors, non-alloimmunized, alloimmunized don't care, so there is anti-TLR4 there or not. So basically this is happening, this regulation of CD83 is occurring in response to heme, is occurring through TLR4. So just another thing, that the toll-like receptor signaling ends in NF-kappa B. This is just the pathway, and the NF-kappa B then induces a bunch of pro-inflammatory, a number of genes. So, so we ask, does this heme effect down-regulate NF-kappa B transcription factor? And also the reason we went there, because CD83 promoter had a bunch of NF-kappa B binding sites, so we were curious to do that. And basically, there are 5 different subunits of NF-kappa B. Interestingly, a couple of them, P50 and RLB, there were differences between alloimmunized and non-alloimmunized in response to heme. Basically, the non-alloimmunized, they are down-regulating the P50 and RLB in response to heme, whereas alloimmunized don't. So this is basically our model of what's going on, what we think is going on based on the data. We think that usually during maturation of dendritic cell, there's a signal through TLR, toll-like receptor ligands, that goes all the way to CD83, say mature, get matured, and then CD83 gets expressed, and now the naive T cell is able to become a Th1. Okay, so the heme, what we think in non-alloimmunized, in healthy donors, may never come across heme, but let's say in a non-alloimmunized sickle patient, and when I mean this is all in the sickle background, right? In the present, you know, when we have a heme signal coming, somehow, we don't know how, somehow it interferes, right, with this signal, so that the CD83 is not upregulated. Now the naive T cell cannot become, or is, you know, basically is saying, calm down, don't become a Th1 at a reduced level, so that you are at a lower risk of alloimmunization. In alloimmunized patients, this signal somehow doesn't work. We don't know where it is exactly, but it doesn't work. As a result, you have a lot of CD83 that basically more pro-inflammatory, the patient is at a higher risk of alloimmunization, okay? So this is where we are. This is just an example of how we're going about it. We're going down, of course, this is it's not a prospective study where we've seen a non-alloimmunized patient become alloimmunized. That's the gold standard, that's where we're going. But at least by identifying some of this, if we can identify the exact place where this signaling goes array between these two extremes, a non-alloimmunized and non-alloimmunized, we can go back and test if we identify exactly which genes there are. The one that Narek was talking about, a polygenic, bunch of genes. If we can identify where they are, this could be one. But by studying it this way, we can then get a handle and then go back and test it in a bigger population before the patients start getting transfused. I mean, that's a whole, but this is where we are. And it was important is just to give you a picture of the kind of stuff we're doing. This is a kind of an interesting story. So let me quickly switch to my second topic, which is patrolling monocytes. So these patrolling monocytes are enriched in this CD16 high circulating monocyte in humans. And what they do, they crawl along the vasculature. And they phagocytose cellular debris from dying cells, dying endothelial or damaged endothelial cells. So they're cleaning the vasculature. They are letting the blood flow freely. I don't know, I think of it as this wonderful, they're working away, they're cleaning, they're keeping everything nice and tidy. And in sickle patients, and they've been shown in mouse models and all that, but in different models, in sickle cell, we've shown that actually the CD16 high population express really high levels of HO1, that heme oxygenase 1, and they control T cell anti-inflammatory profiles. And what we found was that in looking at it more closely, that these patrolling monocytes for sickle patients, only in sickle patients, not in healthy donors, no one else, has this extra subpopulation, this really high HO1 high subpopulation. And we've shown it's a subpopulation because we did some comparison, it's all published, but as many as, in some patients, 60% of their patrolling monocyte express this this HO1 high population. Again, unique to sickle patients. And then we've also shown, I don't have time to go into it, but this HO, where is it coming from? It's not just because if you mix monocytes with heme, you get this. No, you need where it's coming from is from basically uptake of heme damaged endothelial cells. This is really important. This is where it's coming from. So we hypothesize this HO1 expressing patrolling monocyte would be removing heme damaged endothelial cells and possibly even sickle RBC attached to the endothelial cells in sickle cell, dampening inflammation and maybe even, vaso-occlusion, who knows? So this is how we started off. The first thing we had to do is to show that we asked the question, are these patrolling are capable of picking up red blood cells, okay? And what we did, we looked at sickle patients and healthy donors, and we only saw it in sickle patients and only in their patrolling monocytes. And this is tabulated here. And interestingly, it was when patients were going this pain crisis, where there is more hemolysis actually, what happened was there was more of these GPA. So the way we looked at it, we did intracellular stain for glycophorin A, okay, which is a red cell marker. Anyway, so there's more of that. On the other side, we saw there were less of these patrolling monocyte in sickle patients, and it was even less so during crisis. So it could be that they're picking these up, these red cells up, and actually this could lead to their death as well. So to actually make sure this was going on, we had to revert to a mouse model. What we did, we took wild type mice now, we gave them fluorescently labeled sickle red cells or control red cells, then hit these mice with heme, and this we had shown previously, it activates the endothelium. And then we looked at, do these red cells get picked up and by whom? And we focused on patrolling monocytes. Actually, we didn't see anything in neutrophils, but anyway, this is patrolling monocytes. We see them only when the mice are transfused with sickle red cells, not control cells, and not in, well, less so in their in the classical monocyte, but it was really mainly in the petroleum monocyte. Then again, I don't have time, it's in the paper that we published. Basically, we looked at what are the interactions, what are the molecules involved. It looks like it's the CD11A, CD18, this is the LFA1, and ICAM1 axis that is really the critical, seems to be the critical one for this uptake by petroleum monocyte. So, you know, this whole thing came down to, can we use these petroleum monocyte? I'm really excited about this data, and I just stuck this in, is that, can we use this petroleum monocyte to prevent vasoocclusion? Okay, so this is the big story, right? Since they are cleaning up and everything. So we came across this drug, MDP, and it's a long name, I can tell you more about it, that if we injected them into mice to increase their numbers, it had been shown. We also knew that low-dose clondronate specifically gets rid of the petroleum monocytes, not macrophages or anything like that, just that. And so we did that. We increased or decreased the amount of petroleum monocyte. Again, I don't have time to go into it, but just to say that these are liver sections. Green is the endothelium, red is the red cells. And this is just the sickle mice, right? With these two treatments, you see a lot less red cells in the MDP treated mice, a lot more red in clondronate treated mice. And then looking at it by H&E staining, these sections, what we found was a lot of infarcts in the clondronate treated mice and a lot less in the MDP compared to control. These are the tabulation, red cell stasis here, a lot more with the clondronate, and then with MDP slightly less. We're trying to, you know, improve this conditions to really get rid of the stasis. We have some, anyway, I don't want to go into, but anyway, exciting data for us. So what are we thinking was going on? We have the endothelial cells in a sickle patient. It damages the underlying endothelial cells because of the hemolysis. Now the endothelium acts like a Velcro. Red cells are attached, petroleum monocytes come, and through a number of molecule interactions, gobbles up. As a result, we've shown HO1 goes up, and that's cryoprotective. I didn't go into it, but that's what it's doing, is preventing its death. This is trying desperately to keep the petroleum monocytes alive. And this is when we don't have enough petroleum monocyte, which is during vaso-occlusive crisis, we saw the numbers going down. What we find is that now the problem happens. There is a lot more attachment to the endothelium. Now you start bringing in the neutrophils, the classical monocytes, and that leads to vaso-occlusion. Transfusions, and I will have to come back to tell you about that, really improve the life, the survival of these petroleum monocytes. So they're good. They are good for these sickle patients. They are really improving the lives of these petroleum monocytes to clean the vasculature. And I'll leave you at that. I really have to thank all the people. Yongfen Liu is a postdoctoral fellow who did much of the work, and Emmanuel was a old member of the lab who did the work. I'm very grateful. Just like Dana said, we just received a big grant from NHLBI, and we have now a New York Blood Center Sickle Cell Transfusion Research Group. These are the members. And of course, all of this, I'm grateful to our clinical collaborators. For this grant, we are working closely with the Montefiore Health Center, Deepa Manwani, Katerina Manetti, and Joan Eulinger's group. And previously, we worked with CHOP, with Stella, and Dave. And again, I'm really grateful to NHLBI for their support of our program. Thank you.
Thank you very much, Karina. Really, really interesting work that you're up to. This presentation is open for questions, so you could put your questions in the chat room or take yourself off mute and ask the questions. I see Dr. Conway with a hand up.
Unmuted. That was fabulous. I had a question. Tissue factor, being a clotter and tissue factor playing a role in sickle cell disease, is it expressed by those monocytes, those HO1 positive monocytes, and is it regulated by HO1?
Right. We need to work together. I don't know. We haven't looked at tissue factor. I keep away from coagulants. No, but I don't know. I honestly, I don't know. But this is, it's come up before, but we just haven't. I'm so sorry. Yeah. But you know, that would be an idea. Who is it? I thought it was more on the classical monocyte, not on these guys. But I you know, it's my understanding. The tissue factor is more. I don't, you know, it's so context dependent, right? An immune cell could be good or bad depending on its context. It's so annoying. So, but I, you know, so what I'm trying to say is tissue factor, because of their coagulation, I think it's more, we're finding more and more that the classical monocyte are playing a really nasty role. I see everything as a film, you know, these are the bad guys and these are the good guys. So this is how I explain things to my child. So so I think it's more on them, but we need to look at it. And yeah.
Good.
Great, thank you, Karina. Anyone have other questions? I was just sort of in my own head thinking about the role of transfusion in sickle cell disease, and the fact that we're normally taking all of the leukocytes out on filters. Do you think that we're removing a population of cells that would otherwise be helpful?
Right. So very good question. And, you know, if there was a way, and this is my dream, if there was a way to enrich, as I said, you know, enrich for these patrolling monocyte, it was a way, and then give it back to the patient somehow through the, you know, whatever, I don't know. So that would be great. If we could either keep that, just the patrolling monocyte in the unit, or get it out and give it, and, you know, we keep toying with that idea that we're going to do it, but we can do it together. So
yeah, great.
Okay.
Thanks, Karina.
Okay.
There's got one question here in the chat. Is, are you exploring effects of exogenous carboxyhemoglobin that is anti-vasoconstrictive hemoglobin-based oxygen carriers that are used in some treatments?
I think that's a great, thanks so much for that question. So Christine, hi. So what I think, we are not, just quickly, but you know, like conceptually, yes, this could be a way to increase maybe HO1, and you can kind of think of it that way. You know, it's weird, you know, hemolysis could be good, you know, in a way, because it's upregulating in HO1. But the problem on the downside is it's also killing, right? It's a balance. It's so annoying. It's really problematic. So I think, I'm sorry, Christy, I don't know how to answer your, you know, it is based, you know, like exactly what you said. It could have a good effect in these patients, and I think that that's being done, or they're showing that. But going into the mechanism and how it works with these cells, I'm not sure which way it's going to go, and we would love to test that as well, if there was a way, you know, in our systems at least. Yeah. I'm not answering your question, Christine. I don't know. So this is yet another I don't know. but it's working well for these. I think that was the whole idea, right? To use them. I still love Christine. Thanks.
Great. Okay. Well, thank you very much, Karina. I really appreciate the talk. Really interesting stuff.
Thank you so much. Thanks so much.
Okay. Bye. So our last talk before our break is going to be given by Dr. Mel Krajden, who certainly, if you're in Vancouver, needs no introduction. Mel is the medical director of the British Columbia's Public Health Laboratory and the head of hepatitis at the British Columbia Center for Disease Control. Mel's also a professor in our Department of Pathology and Laboratory Medicine at UBC. Mel did his medical training at McGill and then completed fellowships in both microbiology and in infectious diseases before he became an academic, which he started out in Toronto and then saw the light and came to the West Coast. I think, as many of you know, Mel has been central to helping British Columbia build a global reputation in the field of communicable disease control. And he was most recently recognized for all of his very profound contributions in this area by receiving the Order of British Columbia, this summer. Mel also, I think Ed and I figured out, Mel holds the record for the most presentations at Center for Blood Research Symposia, because he always comes and talks to us every time there's a viral outbreak that we're exposed to. And so that's no different today. Mel is going to talk to us about COVID-19, and Dr. Conway is going to organize the slides. Ed, you're on mute. I'm unmuting you. No, I'm not.
Right, Mel, you good?
Yep, you're unmuted.
I think I'm good.
You look great, Mel.
I need to see the slides. First of all, thank you very much for that introduction, and I'm humbled to be invited to come back. So let me try and get rid of.
Hang on, can you see the slides? Did I share or not? No, I didn't share. That might be why.
Share.
Hang on, that's me. Share. Okay. We're good. How's that?
Okay, except I don't see them, so hold on.
Want me to do it for you?
No, let me see. Oh, perfect. Okay, we're done. We're done. Good. Okay, so Ed titled this something COVID-19, and I found this term called bathed in fear in the sun a couple of weeks ago, and that represents, and you heard a little bit about that from Graham Scher, everything is canceled, stay home, go, and that's part of the world we now live in. Ed, next slide. So just in terms of very quick background, we all know that COVID-19 outcomes profoundly increase with age and underlying comorbidities, particularly if you're over 70. If you think about host genomics, there'll be correlations with HLA and other things, and angiotensin enzyme expression. But fundamentally, the profound correlations are with an aging genome, and someone's going to have to research that. If you look at viral genomics, the virus mutates slowly, probably 2 mutations a month. It's enough to monitor global transmission or regional transmission. But there is some literature about differences in virulence and infectivity, which is really incomplete, I think. There's limited therapeutics. I won't discuss that. Next slide. We know that we're in a vaccine race, and I'll talk about that. What you see is the challenges in getting public policy correct. It's difficult. And BC was an early winner, and that typically involves a collaboration between science and politics. But no jurisdiction has been immune to this virus, depending on the political stripes. The next important thing is the unintended consequences are costly. So income, food security, mental health, and ultimately the poor, the vulnerable ethnic populations who work more closely together, live in confined spaces or in congregate settings and use public transit, they're disproportionately affected. And what we realize is that science alone can't address the socioeconomic and racial inequities that are being exposed by COVID-19. Next slide. So what I wanted to do is talk a little bit about transmission and PPE, largely so you understand some of the challenges. Talk about nucleic acid testing, genomics and serology, living with COVID-19 and its relative risks, and some vaccine challenges. Next slide. So if you look at the roots of SARS-CoV transmission, yes, you can find it in the urine, yes, you can find it in feces, but if you're the person on the left and you're an infected host, the major sources of transmission are felt to be contact or droplet, and typically these are droplets greater than 5 micrometers, and these typically don't travel more than a meter. You can get aerosols. These are smaller, these are airborne, and we'll talk a little bit about these nuances. So if you're the susceptible host, the major points of entry are eyes, nose, and mouth. And there's this literature antedating COVID, which showed that typically people will poke their nose during conversations like this and meetings between 15 to 25 times an hour. And if you touch your nose, it goes directly into your lacrimal duct, into your nasopharynx, and it's a good source of getting infected. Next slide. So on this slide, you know, there's this thing about transmission risks and 2 meters apart. And Chu in the Lancet did an excellent summary of the literature. And you see on this graph the distance in meters and the absolute risk. And essentially, it's particularly for this high baseline risk where if you're in a confined space, you tend to have some risks if you're under 2 meters, and that risk goes up the shorter the distance. And for most situations, 1 meter may be sufficient, and the challenge is the public health messaging. So in this review, if you look at COVID, SARS-1, or MERS, physical distance matters, but airflow and dispersion are important. And the best solution for pollution is dilution. So you can imagine that if you're outside, you don't have the same things, concerns about droplets or even spread of aerosols if you're in a confined space. Those things can sit around. The other thing is that face masks are important, and there's a gradation in mask quality. I'll talk about that in the next slide. And then one thing is, if you wear a face mask properly, it does prevent you from picking your nose, depending on the mask. And you'll see that it affects the dose. And then finally, eye protection is important. And where they've looked at this, the use of masks and eye protection are very, very potent protectors of the individual and transmission. Next slide. So this is a paper from Fisher from Science Advances, and it's not a perfect paper, but what it shows is the relative droplet count and transmission through various types of face masks during speech. In that red, in the green area, there's if you don't have anything, and then fleece is not very effective. And you see on the left that a fitted N95 is the best, but a surgical mask is very close to that. And then you see this graded benefit of various masks, and you can see that fundamentally the risk of transmitting a droplet or receiving a droplet is possible when you're using a mask. And you could argue about the efficacy, but a mask is better than no mask. Next slide. In terms of environmental stability, there's a publication by Dor Malden from New England Journal, which looks at the predicted decay of virus from aerosols on copper, cardboard, stainless steel, plastics for SARS-1 and SARS-2, and also the half-life of viable virus. And while certain surfaces are better in decay, like copper, because it tends to be bacteriostatic or microbostatic, the reality is that most surfaces, is not an important fomite. So in other words, yes, we're worried about environmental cleaning, but the literature is that if you have a moist surface, you're more likely to transmit a higher viral load. And if it's a dry surface, it's unlikely to transmit virus. Next slide. So on the left, there's this let's explain it for dummies slide, which I think is actually good. Do face masks make sense? If we all run around naked, and I think we're talking about males here, and someone pees on you, you get wet right away. Okay, if you're wearing pants, then some pee will get through, but not as much, so you're better protected. That's a pretty good analogy. And then below, if the guy who pees is also wearing pants, then the pee stays with him and you do not get wet. So this is a male sort of kind of thing, but it simplifies what the challenge is in terms of masks. And I think that the evidence is overwhelming that masks do prevent transmission. And then, of course, it's important to use the mask. This isn't the mask you wore to school this morning. no, this one is way cooler. I traded mine to Taylor, who traded with Hunter. So trading masks isn't a good idea. And then I have this little microbe washing their hands. And the reality is it's the combination of masks, eye protection, and hand washing that really works. And the data is that if you use PPE properly, it works. And for people, for example, even working in a hospital setting where there are people who are infected, if they wear PPE properly, their risks are more from their household than from their work setting. So this is an important thing that I think people need to understand as we get bathed in fear in our response to COVID. Next slide. So this is a very interesting paper by Gandhi, recently published in the Journal of Internal Medicine, and it talks about infectious dose. And the notion here is masks do more than protect others during COVID because they reduce the inoculum of the virus, and that likely protects the wearer. And it reports data from the Diamond Princess, which was docked in Japan, 634 people. They were tested, and 18% of their infections were asymptomatic. And then it compared it to an Argentinian cruise ship where they had 128 people on board. And in that ship, 81% were asymptomatic. And the difference was in the Argentinian ship, surgical masks were issued to all passengers and N95s to all staff as soon as the first person got sick. Similarly, there had been studies in the Oregon seafood processing plant where they were required to wear masks, and that most of their infections were asymptomatic. Same thing for the Tyson chicken processing plant. And so this notion of a mask on the person preventing the person from transmitting to someone else and to receiving infections suggests that there is a relationship between masks, transmission, infectious dose, and symptomatic disease. And this is akin to with chickenpox, where you'd have your first kid who had chickenpox. This was before the vaccine era. And then they'd come home and infect a sibling, and they would have much worse disease because their infectious dose was much higher. Next slide. So this is an interesting slide from the Winton Center for Risk and Evidence from Cambridge University. And on the left, females, on the right, males, and it really tracks the 5-year average mortality for the period and reports on deaths per 100,000 as a function of age. And what it shows in red for females and blue for males is there's an exponential increase with age that is roughly proportional to the normal death rates, but these are in addition to the normal death rates. And really where you see those increases is where you start getting over age 45. And I've drawn circles around younger individuals where you see very, very little impact. And this is important as we try to get kids back to school, because kids who are not in school, particularly the socially disadvantaged ones, are severely disadvantaged. Next slide. So there's another study that I'm showing here, 2 studies actually. So one of them is the data that children, these are not teenagers, but these are children, tend not to spread their infection. And on the left, you see the Dutch study where you essentially have age of the person who infects and age of the infected person. And you see this co-alignment. And then if you look on the circle, you see that very few infections occur in the very young. And often you can see in some of the Chinese literature that it was the parents that infected the children rather than children infecting parents, which is much more common for something like influenza. And then in BC, we have shown similar data where you have the correlation between age of primary case versus age of secondary case. And again, the circle shows that you have a dearth of cases in the very young. And there's some biases because you don't always test the very young, but it's an important issue as we try to bring kids back to school. Next slide. In terms of the diagnostic mainstay, that's based on nucleic acid testing. And with the tests that we have in Canada, we think that overall you're looking at about a 90 to 95% sensitivity and about a 99.5% specificity. And it's basically detecting viral RNA. The nasopharyngeal swab is optimum, certainly for an outpatient. It's a little more sensitive than throat swabs, but there have been these global swab challenges in terms of supply chain, and people are using saliva, which is a little less sensitive, and we're in the process of distributing saline gargles, which seem to be as good as the NP swabs. It's important to turn around the test quickly, so turnaround times of less than 24 to 48 hours helps people know that they're infected and can reduce the transmission. You can miss some infections, and about 1 in 200 will be false positive. Most of those false positives are analytical or technical. The tests themselves don't cross-react with any other viruses. So because I do believe most transmission relates to symptomatic infections, and we'll talk about this, but pre-symptomatic and asymptomatic infections occur, and I've just mentioned that. So you have this aura of stealth infections, and that scares everybody. And one of the challenges we face as a society is to use testing as a mechanism to open up businesses. In the US, in the Lancet, they've recommended that 4.3 million US citizens be tested every day. That would translate to 430,000 Canadians every day, which is just an astronomical challenge. There are these new antigen tests that probably are about 4 logs less sensitive than nucleic acid testing, maybe 2 to 3 logs less sensitive. And the challenge is they can be done quickly and may correlate with more active infection and might work if you have a cluster of infections where you can use their incremental sensitivity. Next slide. So this is a very interesting a slide from Nature Medicine, where they looked at a receiver-operator curve on slide B and slide C, figures B and C, which really looked at symptomatic individuals in the UK and in the US, and basically showed that the area under the receiver-operator curve is about 75%. So in symptomatic people, there is a fairly good correlation between detecting a NAT positive sample. Next slide. So one of the challenges, and I alluded to it a few moments ago, is if you look at the log viral load on the left, and you look at the days since exposure on the x-axis, what you see is you can try to have the perfect test, or do you use a rapid test that's less sensitive but works faster? And then the challenge is getting the results back to the person. I can tell you that this is a colossal nightmare in terms of delivering sufficient tests so that the turnaround times are good, and so you can actually plan a safe work environment, safe school environment, safe healthcare environment, and this is a challenge. Next slide. So a little word on genomics. So essentially, there's been thousands and thousands of the virus sequenced. We've sequenced almost 2,000 of them already. And what the whole genome sequencing allows you to do is to differentiate ongoing community transmission from repeated introductions of new strains, or to understand multifocal transmission clusters. This is an old slide. The work's been done by Richard Harrigan and Natalie Prestojecki. And what you see is that in BC, the B1 strain, which is this green strain, they're the dominant lineage, as is the A1 strain, which is in that turquoise, not turquoise, I guess, mauve red color. And what's interesting is that the B1 lineage correlates with European strains, and the A1 strain correlates more with US strains. And the challenge, if you look at the next slide, is if you look at the genomics, so there was a very interesting paper by Korber published in Cell recently, where you've seen a shift globally from the D614 to the G614. And this is a mutant that occurs in the spike. And you see this global shift. And if you read that paper, it says, well, there's this shift. And is this because of the transmission dynamics or a fitness advantage of this particular mutant? And they suggest that it's associated with higher viral loads and it's more infectious and less lethal. But the reality is that when China shut down its infections and you had these transmissions from Europe and the US, you really have these biased data sets where you really don't know whether this is just natural evolution of the virus versus an important issue. There's some suggestion that it's easier to neutralize, and the genomics of this virus will be important when you're planning the vaccines, because if it modifies the neutralizing epitopes, that will be important. There was another paper just recently published by Young in The Lancet, and it purports that this Delta 382 deletion variant is less virulent, and those infected had milder disease. But if you look at the paper, what you see is those people affected with the mutant were much younger. And I've already shown you that the relationship to the outcome is largely dependent on age. So it just is an illustration of how easy it is to get something published. And then the question is how do you interpret this very complex data? Next slide. So in terms of serology, so there was this hope that serology would give everybody an immune passport. And in summary, basically, commercial lab-based tests are probably about 95% sensitive, probably 99.5% specific. There are some point-of-care tests that seem to be a little less sensitive, but they tend to have inconsistent quality, so people haven't been using them except in some jurisdictions. If you look at the uses of serology, what's been shown is that it actually has limited diagnostic utility. And the reason is that it takes usually 2 to 7 days for most people to get diagnosed through an exposure and being NAT positive. But it takes serology, about 30% will be positive at day 7, 72% by 8 to 12, 91% by 15 to 21 days, and about 95% by day 21 to 35. And this is summarized in a Cochrane database review. And what it means is that you need the nucleic acid test to diagnose them. The serology, by the time it's positive, you're usually not very infectious by day 9 anyways. It is useful for these multi-system inflammatory syndrome diagnostics. That's like a Kawasaki's disease. And typically those kids are either nucleic acid positive or serology positive at a later point. It's been used to assess seroprotection in rhesus macaques. There's some evidence that you're protected, but as of yesterday, there were at least 4 human reinfections in people who were shown to be seropositive. And the most important thing, and this was alluded to by Graham this morning, is its use to understand seroprevalence. Next slide. So if you looked at other human coronaviruses, there's good evidence that antibody and cell-mediated correlates of protection exist. And the big question is, what is the durability in relation to COVID-19? And I've mentioned that there are reinfections. And so you're sort of stuck at this time that based on the scientific evidence, you don't have evidence to alter the distancing. You don't have evidence to change the personal protective equipment. And this notion of issuing an immunity passport, that passport may not be worth the paper it's written on. One of the challenges in terms of seroprevalence, and if you look at serotracker.com, which was funded by the Federal Immunity Task Force, what it shows is it reports published literature on seroprevalence studies, and this was the report as of September 4th. And what you see, and Canada's down there with studies that suggest that there are 1, and Graham mentioned that the recent data from Hema Quebec and CBS highlights that the overall prevalence in blood donors, which are not totally representative, is a few percent. And the reality is, in different jurisdictions in the US, it ranges from less than 1% to about 10%. And in certain subpopulations, it may be very high, and some illustrations may be in Iran and Switzerland. But the most important thing is in those heavily impacted areas, many more people have not been exposed to this virus, and therefore they are subject to second waves of this virus. Next slide. So in terms of herd immunity, and there was this paper in Science by Britton et al., which suggested that on the basis of an R0, the R0 is if one person is infected, how many other people will be infected in a non-immune population? And most people believe that this virus has an R0 of around 2.5. And they looked at it as a function of age and communities and mixing based on social activity. And they came up with a herd immunity level of 43%. And if you look at most of the other published literature, they suggest 60% of the population need to be infected. And the implications are if 60% or 43% are infected, then every person who's infected is very unlikely to transmit their infection to somebody else. And one of the challenges we face is how do you measure this? Is it antibodies? Is it cell-mediated immunity? There was some publication from the Karolinska Institute suggesting that CMI may be better to measure, but the reality is that CMI is harder to measure than antibody. Next slide. So if you look at all of this complex literature, what do we project for SARS-CoV transmission in the future? Without a vaccine, we expect upcoming waves. And those waves will depend on multiple factors, including control measures. And you can see the politics of control measures differ across the world. We can expect at least another year, 2 years of significant activity with periodic hotspots in these diverse geographies, largely because there's a lot of people, there's a lot of fuel for this virus to burn through and infect others. And normally, if you look at the data for coronaviruses, they're seasonal. But in fact, what's happening is that this virus, because it's just come into the community, the global community, it won't become a seasonal virus until years from now. So that's an interesting factor as well. Next slide. So one of the things that is reported often by Bonnie Henry in British Columbia is a look at what's going on in BC. And you can see that we had a good response in April. And then you can see this slow increase that's occurring towards the end of August. This is August 13th. But you can see even in our current data that we are having more transmissions. And these transmissions are predominantly in younger individuals. And we are trying to protect more of the aged individuals who are at greatest risk of dying. And so we're in this time frame where we're about to start our influenza season, and we are trying to get kids back to school, and you're trying to really keep the infections low enough so that you don't overwhelm your hospital systems and get people hospitalized or on ventilators. And so this is the challenge we face. And if you actually look at what went on in Australia, because of control measures, they've had a very quiet influenza season. And so the challenge for us is figuring out what's the right approaches that opens up the economy, gets people back to school, protects people enough so that they feel comfortable, and at the same time, not overwhelm your healthcare system. Next slide. So vaccines. This vaccine stuff is fascinating. So there's tons of vaccine platforms, DNA vaccines, RNA vaccines, recombinant protein vaccines. There's viral vector ones that don't replicate, viral vector ones that do replicate, so they can actually pump out more stuff. And there's even attenuated virus things. And this paper by Funk is an interesting analysis, but you can see it's published recently and it's already out of date. If you want to see where we are, if you look up the London School of Hygiene and Tropical Medicine, as of September 7th, there were 239 vaccine candidates and 40 in clinical testing. And if you're watching, the AstraZeneca Oxford vaccine is on hold because of a side effect that they're going to investigate. So this is an interesting challenge we face. Next slide. And I think the challenge I think people have to understand is normally to make a vaccine to a new agent typically takes 5 to 10 years. And it involves testing which antigens, testing the adjuvants, testing in animals. You can see that you have these phase 1, phase 2, phase 3 trials. And then you still have this phase 4, which is what happens afterwards. So if you looked at the dengue vaccine, it worked, it took a long time, but when you had it in phase 4, you saw that it actually had bad side effects. And so here you have this totally compressed environment that we're supposed to have a vaccine in the US by November 1st. Certain president said that it's going to be in place. But you can see the challenges faced by industry to try and rapidly respond to a virus where we don't fully understand the immunity. Next slide. So here you have Vladimir Putin riding this bear with a big syringe on his back, and he's going to solve the COVID, and he came off the first on this. And then our packaging design is like our vaccine. There are 0 corners, we cut them all. And then on the other hand, you've heard of Operation Warp Speed, which there's a very good editorial on its strategy, which is actually an outstanding strategy from the US. But of course, you have the political component. And so I think that someone's going to come up with a vaccine that does intradermal injections, and it will be part of a tattoo session. So maybe that could help us improve what's going on. Next slide. So here's, I think, one of the important things for us to think about in terms of conclusions. So infection will lead to some protective immunity. We don't know what that duration is, and we are going to all work on coming up with the antibody and CMI tools to measure it. If achieving herd immunity requires 40 to 60%, even in the highly affected areas, there are many more to affect. We're going to have more waves, and we're going to have to figure out how to live with that. The unintended consequences of our response are severe, and you need to have civil societal discussions. And it actually is a two-pronged thing. It's a civil conversation, but a civil societal conversation to come up with acceptable approaches. And you can see the controversy that occurs in different jurisdictions, the juxtaposition of rights that are in individual versus public. A fast-track vaccine needs to be safe, and the reality is that's going to take time. And vaccines don't often work well in the elderly, and they're the people who are most effective in terms of a bad outcome. I think one of the most important things for us to remember is there are consequences to life in bubbles. So if you have a gnotobiotic animal devoid of being exposed to bacterial or microbes, they don't develop an immune system. And so our immune system works on a use it or lose it principle. So the notion that we can live in bubbles for the rest of our lives is fraught with a problem. So I think fundamentally I should stop there and open for questions. And with this last slide, I will say COVID-19 is the pandemic's pandemic. Take everything I say with a grain of salt. Thank you.
Thank you very much, Mel. Really fascinating story and where we are in this particular journey. There are quite a few questions in the chat. We may not get to all of them because we're a bit behind on timing, but maybe I'll just start with this one: is what impact does the low prevalence of SARS-CoV-2 have on the positive predictive value of tests being performed? particularly in asymptomatic people? And what are the implications of this on test interpretation, especially in those asymptomatic individuals?
Yeah, so it's a perfect example. So we have, as a province, shied away from asymptomatic routine testing. And what we have shown is that in some of these minimally symptomatic cases, you have false positives. And so 1 out of 200 people is going to be a false positive for a variety of technical reasons. And that has a massive impact, including the fact that if you want to go traveling on a plane or traveling to a country, someone has to test you. And the argument is that it consumes public health resources to try and chase after these people. But I've also shown you that some people get in asymptomatic infections. But this notion of blanketly testing everybody, you can't test your way out of it. It has to be a thoughtful approach to trying to do the test that is appropriate in number and scale to help you open up your society, keep your schools safe, keep kids protected, and things like that.
Thank you. And then maybe I'll just take this one last question, is that given that there is evidence that the immune response to SARS-CoV-2 may not last longer than 2 to 3 months, what are your views on the importance of finding reliable treatments versus an effective vaccine before return to normal? For example, from a public health perspective, do we need a vaccine to have the next Norman Bethune Symposium face-to-face, or do you think that good treatments would be satisfactory?
You know, there's a bunch of the debunked treatments. What's clear is remdesivir isn't as good as people thought it was. You can use the dexamethasone and steroids in severe disease. They are working on treatments. But if you look at most viral illnesses, it is very hard, unless you have a treatment that can go into the water supply, to actually reach the people that need to be reached. So for a viral kind of illness where you can have this transmission, the best solution is a vaccine. I would also say, just because your antibodies decline, we don't really understand the immunity correlates. It may be that the antibodies to the N gene decline, spike may decline, but overall there may be immunity that actually interacts with other human coronaviruses. So I don't know what the answer is, but I don't think you can just solve this by just creating therapeutics.